Principles of Bioproduct Separation, Module 14, Ancillary Operations, Segment 1, Water Quality. With me, Yusuf from School of Life Sciences and Ethnology, Institut Teknologi Bandung. In this module, we will discuss about water quality, solvent recovery, waste disposal, and last but not least, biosafety. Ancillary operations are other operations that do not appear in the main sequence but are essential in bioseparation. Ancillary operations serve two basic functions. First, they maintain an adequate and economical supply of raw materials for the mainstream processing. Second, they protect the environment, personnel, and product from contamination. Water quality. An adequate water supply of appropriate quality is essential for various streams in bioseparation. There are two important aspects in water quality: microbial control and chemical control. Microorganisms can easily propagate once in contact with the generally organic-rich process stream and can cause product deterioration. Microbial control must be especially stringent if the product is to be injected. Hence, all pyrogens have to be removed or otherwise may cause fever when injected to human and animal. Unlike microbial cells, chemicals in water do not proliferate. However, a minute component can cause a major lowering in product quality. Any contaminating chemical can have three possible fates. First, it can be separated from the product. Second, it can be co-purified and concentrated with the product. And third, it can be unaffected by the recovery. Water is grossly classified into four levels. Class 1, often referred to as well water. It is used primarily for utilities. Class 2 may come from a municipal water supply or from a private well for drinking water. The water is chlorinated at its source, but the level of chlorine and microbial contamination vary. Usually, the treatment includes deionization and filtration for particulates removal and widely used in bulk chemical processing. Class 3, classified as purified in US Pharmacopedia, prepared by deionization followed by ultrafiltration. It can contain no added substance, thus no chlorine is present and consequently microbial control is difficult. Without the presence of chlorine, it is usually necessary to treat the stored water for microbial control. Sometimes a recirculation loop is installed in the storage facility, and the system is maintained at elevated temperature, 80 degrees C, to minimize microbial colonization. Class 4. It has the most stringent of quality standards and largely used for injection. It must contain no pyrogens as determined by a laboratory rabbit test. Pyrogens may be removed by distillation, which is expensive. They can also be removed by ultrafiltration, but the effectiveness varies. For the next segment, we will discuss about solvent recovery. Until then, I am Yusuf from Institut Teknologi Bandung.